Okay then. Right, so welcome everyone to the webinar series titled Stylish Academic Writing. The webinar is organized by Education and Training Committee under the International HUPO Program Organization. My name is Justina Verbober. I am from the Cedars-Sinai, California, and today I am your host. Today is also going to join me Tiana Guo from the Westlake University from Shanghai and Brian Surley from the Ohio State University. The goal of the webinar series is to increase student agency as writers and to provide them and explain the tool of sets that they can use and train in order to help them with the critical thinking, writing, and reading. So it is my great pleasure to introduce our speaker, Professor John Yates. So Professor John Yates is a chief of the journal editor, Journal of the Proteomics Research. And we thought that he will be the great candidate to start our webinar series. So his research interest includes development of integrated methods for tandem mass spectrometry analysis of protein structures, bioinformatics using mass spectrometry data, and biological structures involving proteomics. His laboratory developed the use of proteomics techniques to analyze protein complexes, post-translation modifications, organelles, and quantitative analysis of protein expression for the discovery of the new biology. Many of his proteomics approaches has been nationally and internationally helped to many investigators in the scientific community, and he also received multiple awards, including the American Society of Mass Spectrometry Research Award, the HUPO Distinguished Achievements Award in Proteomics, the ACBMA, and Christine Atkins Awards of the Proteins. Professor Yates, why we decided to invite him. So as I mentioned, he's the chief editor of the Journal of the Proteomics Research from 2016. But before that, he was also editor for the Analytical Chemistry. He is also a well-recognized person who published more than 70 scientific articles with the more around something like 70,000 citation. So I think so that's, it doesn't have to, I don't have to tell you more that this is the person who will give you a lot of good points, how to strengthen your writing, how to improve your writing. And I think so without further ado, the title of his talk is Write. So welcome, Professor John Yates.
Thank you. Thank you for the invitation to do this today. I'm coming from rainy San Diego this morning. We're very happy to be getting rain today, finally. So I've got some few comments to start with. Okay, hopefully everybody can see that. I thought I would title this as Write because I think writing is a lot like working out in the gym, where the more you do it, the stronger you get. And with writing, it's the same sort of venture. The more effort you spend on trying to write things, the better you'll get at it. So I thought the title Stylish Academic Writing was a little bit of an oxymoron, because technical writing is usually anything but stylish, although there's no reason why it can't be. So that's certainly something that people can work on. I'm sure you've all read papers that you thought were extremely well written and entertaining, because they're telling a story in part of the writing. So I want to talk about three parts of academic writing. So one is mechanics, the other is style, and then the last part, which will be the longest part, is content. And fortunately, we just put together an editorial in JPR where we talk about what we are looking forward to seeing in research papers, and it's both a reminder of what we expect in papers, as well as just kind of laying out what our views are about what type of papers we'd like to see. Okay, so in terms of mechanics, one of the best things you can do is to read a lot and pay attention to the writing of others. So don't just read to get the content out of it, but also look at how people, particularly if you're reading something that you find enjoyable. I mean, look at how they've constructed things and how they've written the particular article. I would also say, if you struggle with writing, to take a class, especially if English is your second language. There's no reason why you, I mean, that helps with the practicing of writing and so forth. And I encourage my postdocs from other countries where English might not be their second language is to take a class and do that. So, because proper grammar is really important when you're writing a paper, and it can be a deterrent for when a paper is reviewed, as improper grammar can really detract from the content. So if somebody can't understand what you're trying to say, you could have the best scientific content in the world, and people just aren't able to get it because they can't understand what you're trying to, what you mean. There is a course on Coursera, which they have a class on technical writing that is very good. During the pandemic, they were allowing people to take one free course or something like that. And my wife took this class on technical writing, and she thought it was excellent. She thought it really helped with that. And the other thing I would say is get a copy editor. I think Grammarly is now trying to do this online. I've never actually tried it, so I don't know how well it works. But having somebody you can rely on as a copy editor is really helpful, because particularly if that person's not exactly an expert in the topic in which you're writing, because if it's not clear to that person, then it's not going to be clear to anybody else that's going to read it. And my copy editor happens to be my wife, who of course has taken this technical writing, and my first drafts often come back with lots of red ink all over them. And then the last thing I'd say is that, by writing a lot, you can improve the mechanics of your writing, especially if you pay attention to what you're writing. I know that when I write, usually the first draft is more free association than it is a well put together piece. So so you should be going through multiple drafts when you're writing something. So the first draft is just getting words down on paper, and then the second and third draft are getting them better organized and more clear. Okay, second part is style. Again, it's important to read and pay attention to the writing of others so that you can see how people are doing this. And many great novelists agonize over each sentence. Hemingway in particular was a perfectionist when it came to his writing, and he went through many drafts of just trying to rephrase a single sentence when he's writing things. And so the style, you also want to make sure that the sentence say what I wanted to say, and so make sure that that what you're writing, what you're saying is clear. So trying to remember that you're really just telling a story about the science that you've done, and so you want that story to be clear, to be well written, and to make sense. And what can be useful is to get a copy of a book. I've had a copy of this book for probably 50 years, going back to I think maybe high school. which the elements of style. And so if there's some grammatical thing that I'm not clear about, I'll refer to this book to find out what it is. But nowadays you can just Google things too as a way to find out what's the proper way to say something, what's the proper word to use, and so forth. Okay, so last topic is content. And with scientific content, we have a couple of things that we consider when we're putting studies together. So one is novelty. So we always want work to be novel, although that's not entirely true now, because there are journals where novelty and significance is not the overriding factor. So you also want the work to be significant. So just because something has never been done before doesn't mean it really advances the state of the art. But sometimes novelty can kind of override the significance aspect of it, and novelty can just mean something that is incredibly interesting. For example, I had a colleague when I was at the University of Washington. They were having this, I would call it a beer-induced debate about when you take a quarter and you tap it and you can get it to spin. So the question was, does it spin longer in atmosphere or spin longer in a vacuum? And so after they sobered up, they decided that they would try the experiment, and they did the experiment, and you did number of replicates and all that stuff, and they wrote it up, and they submitted it to Nature, and Nature took it because it was not only sort of a novel investigation, but it was there was a lot of human interest in there, even though the significance level of that particular study was probably not going to change the world, but it was interesting. And so sometimes the top journals will take papers where the topics have some human interest nature to them. And the studies also, of course, have to be well done. And there are sets of journals which were triggered by PLOS One, which was one of the first in this class, where they just want studies which are technically competent. They reduce the level of novel and significance, and they want technically competent studies that are maybe advancing something in some fashion. And so there are now a number of these, they call them omega journals or something. Where you can publish these sorts of studies. And so in theory, no matter what you do in the lab, there should be a place to publish it. And one of the things I've learned early on is that publishing is a business, and journals need content. And so there's always a place to publish everything that you've done, things which are not necessarily as novel as they could be or as significant as they could be, but maybe they're just good, well done studies that will advance some aspect of science. So now there are nice places to publish those studies. Okay, so what do we look for at JPR? First and foremost, we continue to prioritize exciting, groundbreaking science. And we have, I don't know how many of you know, but we also publish, and we have for the entire length of the existence of the journal, we published metabolomic science. And in fact, one of our associate editors at JPR is a very famous scientist in the area of metabolomics, so Jeremy Nicholson. And so he's been pushing this side of science in the journal. But we've always expected that at some point, proteomic and metabolomic studies would be done together and to create more omics level science. And so that's been one of the mainstays of JPR over all these years, is that we welcome both types of studies, not just the discovery. So we're looking for lots of different things, software, methods, and so forth, not just the discovery of new biology, new biological mechanisms. And so we're excited to see the development of proteomic metabolomic methods and application to new areas. So I have a fondness for the use of these kind of technologies in a wide variety of different scientific areas. Like we've been publishing papers in paleobiology, archaeology, rare species, the Komodo dragon, and forensic science, to name a few. And in fact, this Komodo dragon study that got published in JPR wound up getting picked up by The Economist, and they published this little blurb in their science and technology section of the journal, The 48 Uses of Dragon's Blood. And those of you who are Harry Potter fans will recognize the play on the play in the title here on some of the Harry Potter movies. Okay, so some of the things that people have been doing over the years is comparing normal states and perturbed states to look for important insights or hypotheses about mechanisms behind the perturbation. And what we're trying to emphasize in this editorial is that when you do these kind of studies, comparing a normal state and a perturbed state, disease state of some kind, and you come up with a list of proteins, those are hypotheses about what might be causing the perturbation, or what might be behind the perturbation. They should be validated and tested in some fashion. For example, you can go in, you can knock out a protein in a wild type cell to see if you can duplicate the phenotype of the disease in the cell type. You can do a knockdown, you can do overexpression, you can do chemical modulation as a way to try to duplicate these phenotypes to try to establish that, yes, of these 10 proteins that we saw going up and down, when we knock down this one or we perturb it in some fashion, we replicate the perturbation and disease state that we originally were studying. And so that goes a long way to making your paper more relevant for those that would come along and try to follow up on this. So while it's interesting to take these quantitative data sets and do standard Go and string analyses, those do not necessarily validate the connection between protein and metabolite changes. They should provide you with more confidence and perhaps better insight on which things to test, but they should not be the final endpoint. So we're also seeing increasing number of proteomic and metabolomic studies in non-model organisms where a genome may not be sequenced, or a genome may be sequenced, but molecular biology tools may not exist for robust follow-up experiments. And for these types of studies, we would remind you that cell lines are closely related to organisms can often be used for follow-up studies. And if you remember back to the rationale behind the Human Genome Project and the genome projects of a number of different other organisms, such as mouse, fruit fly, worm, and yeast, is that those were intended to be used as biological surrogates to study the functions of the related human genes when experiments with humans are not possible. So you can't always go in and do validation tests in people. And so we oftentimes have to use these other surrogate organisms as a way to test things. And they were intended to allow us to ferret out the functions of various genes and so forth. Okay, so we also expect to see proper statistical design of quantitative experiments, and I would refer you to a review published by Olga Vitex and Oberg back in 2009. This was published in JPR, and they provide some guidelines for experimental design and statistical analysis and so forth. And so replicates are critical for determining experimental and biological variation, and studies should include proper statistical, analytical, and biological validation necessary for the study. So we are increasingly looking at the statistical analyses that have been done in the paper to make sure that you've powered the study appropriately and so forth, so that the results are actually robust. And there are certain kinds of experiments where this is particularly important, and those are biomarker studies. So biomarker studies are oftentimes you can't go in and can't go back and validate the biomarker in the same way you might when you're looking at a biological mechanism. So you have to make sure you have enough samples to power the study and provide good statistics. And if when you're doing these studies in humans are lousy experimental subjects because they they are out there living their lives, so you can't control them. You can't control them the same way you might control a model organism in terms of diet and temperature and all those various things. And so this makes biomarker studies all the more difficult because you have to make sure that you have enough numbers to overcome some of the noise that's going to be in the data. So this is especially important. The power and the good statistics, especially important for proteomics and metabolomics, where the number of variables is often high relative to the sample number, and the data is at risk of being overfitted. And so once you've done what we look for in well-done biomarker studies is that once you have a discovery set that's produced some putative biomarkers, you then go in and you validate this in a second set of independent patient samples, and you use receiver-operated characteristic curves to determine the specificity and selectivity of the candidate biomarkers. And we recognize that testing potential biomarkers in a separate patient population can be time-consuming and expensive, that it may not be feasible to test biomarker results in a model system. However, for a molecule to be a true biomarker, rigorous validation is required. And the literature is awash with potential biomarkers, and we want to avoid adding to that noise. And I would suggest that you go into if you are skeptical of this, go into PubMed and type the word biomarker and see how many things you pull back, and it's like 1 million or so papers will come back with the term biomarker. And a biomarker should have a meaning that suggests that it's really been validated and it's useful. If you're working with a rare disease, samples can be hard to obtain, and we understand that. And it's oftentimes that you can improve the likelihood of finding rare markers in these rare diseases by increasing the number of control samples that you're using, which are much easier to obtain. And so controls are critical to rule out false positives that result from overfitting. When working with rare diseases, using twice as many controls as cases can improve the specificity of the markers. So we're not without. We understand that there are lots of issues associated with this. These can be very expensive studies, but I think it's really important that you do them well in order to establish that you've really found a biomarker. So back in the early days of proteomics, my lab did this too, where we would say, pull out a postsynaptic density from neurons, and we'd go in and we'd try to characterize what proteins were present in the postsynaptic density. And back 20 years ago, those were novel experiments, and they've been done ad nauseum since then. But that's not to say that there are not still examples of where you would do these kind of experiments. But as you contemplate these things, we consider these to be data resources. So as you contemplate such studies, keep in mind Sidney Brenner's CAP criteria. And so you want the data set, the data resource to be complete, do a thorough job so that someone doesn't have to come along later and redo it. You want it to be accurate, so take care to ensure the data is well collected and properly vetted, and permanent. The data should stand forever. Now, I think proteomic data is different from genomic data in that it's understood that omics data are dynamic and can change based on conditions, but the data that you collect under the criteria you use should be rock solid. So it should be, the data set hopefully should be written in stone. Okay. Protein lists are also being generated in new and unusual studies, as I mentioned earlier, like paleoproteomics, forensics, and archaeology. And so there have been a number of studies published where people have taken proteins from old skeletons, and they've been used to settle long debated taxonomic questions for extinct animals. I've reviewed a couple of these in some of the big journals, and they're certainly really interesting studies that are helping to settle questions that that having data at the molecular level really helps to solidify what the answers are. So obviously follow-up studies in these type of organisms are not possible, but they're not necessary because these studies are not investigating biological mechanisms. But what you want to do is you want to substantiate that the data that you've collected, so for example, tandem mass spectrometry data, is rock solid. So you want to make sure that you've annotated that data, so you've gone through and you've labeled all the peaks, and so people will be convinced that the spectrum that you have represents faithfully represents the sequence that you've assigned to it, and that piece of information is the information that you're using to settle this taxonomic question. It's also good to substantiate it by synthesizing the relevant peptide or small molecules to compare with the raw data. So you've substantiated that we think it's this sequence, we synthesized it, we've made the tandem mass spectra for it, and it exactly looks like exactly matches the one that we collected on the real experimental system. So that goes a long way to helping to validate and substantiate that your conclusions are accurate. Okay, we also we're actually just finishing up a special issue on software tools for proteomics and metabolomics. We had a really great turnout this year. I think there might be up to 60 papers which are getting published, and so it's a wide variety of tools, a lot for mass spectrometry, but we're also looking for array informatics or tools that help convert data to knowledge. So in my laboratory, where people spend a lot of their time is trying to figure out what's the new biology they discovered from their experiments. And that often involves a lot of software tools that are helping to convert data to knowledge. So we very much encourage the publication of software tools that demonstrate the potential of software to identify new information or improve data analysis performance. We often get papers that describe computational modeling methods to identify drugs or peptides that would be good inhibitors for proteins. These are often just computational studies, but I often push back on them and ask them to go and validate by testing the findings in an experimental setting. In mechanism of action studies, dosing needs to be described and the compound should be well characterized and so forth. So I don't think it's sufficient to just do some computational screen and then say this is here are the top 5 hits, and somebody should go out and make them. I think that people should go out and make them and test them. So JPRS, we've long appreciated and look for methods papers. We consider ourselves to be a analytical methods journal as well. And so we like publishing innovations that help increase sensitivity or throughput or to provide new readouts of protein function, such as conformation, localization, and interactions. So best practices would include showing proof of the method on a system where the answer is known and then demonstrating that it works on an unknown system. We have a special issue on omics methodologies coming up. If you're interested, check out the JPR webpage. Methods should be well described for others to replicate them. That's just a fundamental issue in science, is that you need to have enough information in there that your methods can be replicated. And a lot of people, because of word limits and all that, a lot of times they'll skimp on the methods, but you can always add to your details of your methods by putting them in the supplementary information section. But they should provide compelling advantages over current methods to warrant their adoption. Hence, to support the novelty and usefulness of any new method, it should be benchmarked against the current best method. Okay, we're also we require that data be uploaded into one of the repositories. And so this is not only proteomic data, but also metabolomic data. So there are the proteomic exchange partners like Pride or Massive, and then there are increasing number of local subsidiaries of proteomic exchange. So whatever is most convenient for you, and I wouldn't say that these are the only places where you can put data, but it just needs to be publicly available. And I would, if you can, I would suggest putting data into multiple repositories, because these things are often funded by grants, and grant support can be a little bit iffy. You should also make sure you adequately describe the metadata to support reanalysis and extended analysis of the data. I think proteomics and metabolomics are a very different type of data than genomic data. So with genomic data is once you've sequenced the genome, it's pretty much 99% the same. And then when you sequence other individuals, you're going to see some slight variations here and there. But with proteomic data, the kind of experiment you do, the data can change a lot. And so metadata is really critically important for people to try to reuse data in some fashion. And so make sure you don't skimp on that part of the data. And so there are also places where you can put metabolomic data. There's just GNPS at the University of California, San Diego, Metabolomics Workbench, Metabolites at EBI. And also increasingly people are doing proteogenomics. So don't forget to upload RNA-seq data when you do that as well. And you can also document data workflows for processing and on best practices and put that up on GitHub. So all software ontologies, vocabularies should be described with their version numbers and dependencies and implemented in some form of containerized workflows for reusability. So there are lots of things that you can do to help make your science much more extensible, and that leads to more citations. So when you do science that people can use to move forward, then people wind up citing your work more frequently. Okay, so this is just so if you follow scientific fraud, most scientific fraud tends to come about from Western blot images. So we've required for a number of time, this is just a reminder, the submission of raw, uncropped Western blot images as part of the supplementary information. They should be provided for review at submission. Also, we look for full details of antibodies used, which must be provided to ensure data reproducibility, including species, supplier source, lot number, catalog number, and resource identification number. I actually think that antibodies are one of the prime suspects in the reproducibility problems that we often have in science, is that somebody might get a different batch of antibodies and they no longer work the same way that they worked in the published paper, and then they struggle to be able to reproduce the study. So adding all this information can be really helpful for people understanding why they're unable to reproduce a published study, and it could be because this is a new lot of antibodies and now it's not working as it originally did. So as we tell our laboratories, we're in the business of knowledge generation, so write it up. We look forward to receiving your latest scientific advances, and so all of this will be in a soon to be published editorial, which was signed by all the associate editors at JPR. So I'd be happy to answer any questions, but I would say if you really want to improve your writing, write. It's the best way. It's flexing your writing muscles can make your writing the best. And I would also say that even if English is not your first language, it is possible to be a great writer. One of the best writers to come through my lab was a Chinese national, so Meng Chu Dong. She was she wrote beautifully, but I think she spent a lot of time working on it, and in order to be good at it. I think that's what it takes. It takes a lot of exercise in order to get there.
Thanks, John. There are a lot of great questions already in the Q&A. I could start reading them to you if that's good.
Sure.
So one of the first questions, and I think this is a good one to start with, is from your decades of experience in writing hundreds of papers, how do you know which level of journal is suitable for your work, and how do you balance between submitting the top tier journals and spending time in revisions or resubmissions, or trying to publish in faster journals to avoid getting scooped. I guess I would tack on a secondary question, which is, what scope should a grad student target for their first paper? And by scope, I mean, how big should the science be? And then how would that be different for a postdoc or a junior PI?
Yeah, so if you look at papers that are published in the top tier journals and you kind of do the math of how many years, how many man years, how many person years, let me say, have they spent doing this study? And you probably could come up with some number of like 25 person years is involved in a particular study. And it's probably because they got, 10 people on the paper. So those tend to be pretty large scale studies. And let me give you a proteomic example. So Steve Gidgey's lab has been publishing these protein-protein interaction papers, and he has about 25 people on the paper. And it's taken them a few years to do each paper. And so that's a lot of person years involved in that paper. And so that's sort of one criteria to think about when you're thinking about the paper. But you can also think about it from the standpoint of do the conclusions of this paper significantly advance some aspect of science? And if you think so, then it's probably worth making the effort of submitting to a top tier journal. You'll get an answer very quickly if they agree with you or don't agree with you. They'll either send it out for review or they'll tell you that they're not interested in the paper. And so one of the things that I've learned over my years in science, and so I collaborated, I had part of a P41 center for many years, which is why we have so many publications. And we collaborated with a lot of biologists. And one of the things I learned is that a lot of them will write up their paper and then take a stab at a top tier journal. And if it gets sent out for review, that's great. If they take it, that's even better. If it gets sent out for review and they don't take it, you still get back terrific reviews that help you revise the paper to go to some other journal. And in fact, one of the people in my lab was telling me that I don't know if he was working for this person or he just knew this person when he was at Harvard, and this person would always write up his paper and send it to Nature, and then if they rejected it outright, they would then send it to Science. So what he was trying to do is to get somebody send it out for review, and then he would get back fantastic comments, and so he would revise the paper and then send it to Cell after getting the revisions back. So you can get some people will use the review process as a way to get appropriate critiques of their work to revise it for submission to another top tier journal. So different strategies. I think it's important to understand where the work you're doing fits in with the field and the significance of it and so forth to decide whether to go to top tier journal. So one of the dangers, of course, is that we had a paper that we had submitted to Nature, and it kept going through rounds of review, and it was in review for 2.9 years before it eventually got accepted. But at some point, we got so invested in the process that to stop and go somewhere else, we had just lost so much time, and so we wanted to continue with this, and it just wound up taking too long.
I think so that it's bringing us to the second question, which is, if a project does not turn out to be as great as expected in the middle of purchasing it, it is still worth to be continue and publish, or better to pursue a different project as early as possible to save effort for more important things? Because publishing every paper, low, medium, or high impact, takes a lot of effort, asking from both a student and PI perspective.
I'm in the camp of better to publish than not publish. And I did have one postdoc who published every scrap of data that she collected while she was in the lab. That wound up being like 27 papers or something like that. So it's pretty phenomenal. Yeah, so I do think it's important. I mean, you've spent the money on the research, and so you should publish it, and it might be of value to somebody out there. It's oftentimes hard to anticipate what science is going to be significant. It's not always clear that this study or that study is going to be important down the line.
Yeah, I think so. I strongly agree with you because, as you said at the beginning, yes, the novelty, the significance is important, yes, but also increasing the knowledge about the subject is also very important. So maybe sometimes what you have to repeat, it can also bring some other people more information than you think so, yes. Everything, someone said that everything is important, so.
Yeah, and if you pick the journal right, it may not be that much work. And another anecdote is I had a postdoc, and she'd done a paper that I thought was really good, but she wanted to go to a particular journal, which it was kind of a middling journal, and said, okay. And so she submitted it, and it got accepted within a week. And then she comes back and says, I guess I should have aimed higher. So it may not take as long as you think, and I think it's better to aim higher and then work your way down. And if they send it out for review and you get reviews back, those can be really helpful.
Hi, John. Just now you said you writing a paper like working in a gym, you get your writing skill stronger when you write. Then do you think, let's say we have the same set of data results, and do you think it make a big difference for different people to write it? So after you write so many papers, so what's the most important section you think is make the biggest difference? I think the results probably relatively straightforward. So how about introduction and discussion? Do you think they will, or abstract, do you think they will make a difference for the editor to choose to send to review by Nature Science? Do you think this make a difference?
I like to write the introduction first, because that's a place where I can be more free form, and I can craft the story in the introduction. But that's where I start. And then one of my postdocs, his strategy was to make the figures up before he wrote a word. And so he had to have 4 or 5 figures, and then they all had to have like 3 or 4 panels in them. And he would often say, I'm about ready to start writing this thing. I just need one more panel for my figure 5. And then he would go and he'd start writing it. And so I think that's more of a biological strategy. He was a neurobiologist. And what I would often do when I'm writing the paper is I would start writing the results, and I would write it, and then I would figure out what figure I would want to illustrate some point while I was writing up the results section. So people have different styles. I think it's difficult for multiple people to write a paper together. I think one person, what seems to be successful is one person takes the lead, writes the paper, and then other people start editing it and adding to it. But I think if you, just in terms of actually getting things done, I think it's much more efficient when one person just takes the lead and writes the paper.
Yeah. So now
as much as I can.
Yeah, we have over 2,000 viewers in China. And one question, I mean, it's also actually my question, how to write a very strong discussion. Do you find this difficult? or do you think this is not very important?
No, discussion actually is probably one of the very important sections of the paper, because now you're describing how the results that you've collected are advancing our understanding of what you've discovered, right? So if you've got some new mechanism for autism, so the discussion part is where you're putting your results into context with what is already known in the field and how this is being advanced in some fashion. So the discussion is really where you're bringing all your data together with everything else that's out in the field, and laying out how this is advancing what we now know based on what you've done. So it's an important section.
So in some journal, they like the results and discussion, they combine together. And in some journal, they have a separate discussion section.
Yeah. I tend to do them together, results and discussion, but maybe it's better if they were separated, because then you can, you lay out the results and then you lay out what we now have learned and how this advances what we understand.
John,
But it can depend on what kind of paper you're writing. So if it's a methods paper, the discussion is going to be different than if it's a biological mechanism paper.
yeah,
Thank you.
That makes a lot of sense. One of the other attendees asked a question about, just from a trainee perspective, how many papers should a trainee try to publish and read a year? So like both reading and both publishing, like how much should they focus on reading versus actual research? And then writing as well. Yeah,
I guess it depends on what's going on in their life. How much time they can spend reading and writing. Although they don't always follow my advice, I tell people in my lab that they always should be writing something. And if I had my way, everybody would have some review that they're writing or something, and probably in slow motion, writing a little bit each day, to keep up with it. They're probably not following that advice, but there's a movie that's on in cable rotation right now called My Salinger Year. So J.D. Salinger, who wrote Catcher in the Rye, which is a fairly famous book in the U.S., and he's having this discussion with an aspiring writer, and he says, "Are you writing every day? You should write for 15 minutes every day." That's the process of writing, and so I think people should be writing something every day to keep at it. Set aside some time to write. I like to write. I find writing in the morning is best for me because my head is clear and hasn't been polluted with 50 emails.
So I saw you have quite a lot of editorial in JPR. They're very short and very sharp. May I ask, normally how long do you spend to write each of these editorial?
Yeah.
One morning.
So they take weeks.
Oh, really?
So I'm not working on them constantly. So I will work on them, get it written. I get a draft written, and then it goes to my copy editor, and then the copy editor tears it apart, and then it's back to the drawing board of writing it. So it takes a while because I don't have time to work on them full time. So I'm working on them, an hour here, an hour there.
Do you want to talk about your writing process? Similarly, along those lines, you write in the morning usually?
I try, yeah. Yeah, I like to write in the morning. I get up early. I'm heading into the lab at 6 a.m. And so I find that the early morning hours when nobody's around is the best time to get things like writing done and so forth. And then once people start showing up, the activity level starts increasing, and then it's hard to focus and start concentrating on things. And that's been true my entire career.
Mm.
The other place where I used to do a lot of writing, I don't anymore, was on airplanes. Put on headphones, and I used to write out things longhand before laptops were a thing. You guys are probably too young to remember. But I would write out longhand, and I would come back and type it all in. But nowadays, if you're back in economy, it's hard to open up your laptop to do anything.
Mm. So when you first write a first draft, do you write from the beginning to the end, or do you write one paragraph and then the next day you write a second paragraph?
Yeah, that's something like that. So it's not. So particularly when I'm editing something, I will only print out a page at a time, because I cannot get through multiple pages without going back and typing it back in, typing in the changes. And then, so I tend to edit sections, section at a time. But I do want to be able to see the bigger view of it. So right now I have this 27-inch monitor that I've turned into portrait mode, so I can try to put multiple pages on the screen.
So, so how, as a chief editor of JPR, how you decided what is the main key that you will go and pass this particular manuscript to the reviewers, or you will say, no, thank you, unfortunately, it's not suiting the JPR, but what is the main one? Of course, we are not talking that it's not metabolomics or proteomics data, but what's the main key that you will decide to go green or red?
Yeah, so the first thing I look at is that paper has to be within scope. So it has to fit the journal. So, and then there are some papers which deal with which still might be in scope, but they are, say, more agricultural proteomics. And there's now a journal at ACS that is probably better suited for that area. And so I may decide that's out of scope and send it to that journal. But I look at the science a little bit and decide which editor is appropriate for the paper. Then the associate editor will make the more detailed decisions about it. But I mostly look at scope. If it's if there are any questions, I will read the paper.
So another question, we have a few more questions, but maybe I will start with a question from Ian. And the question is, what is your plan to distinguish JPR from complete with the other proteomics journal like MPC, Analytical Chemistry, Proteomics, Journal of Proteomics?
So I look at JPR as being more analytical proteomics, analytical proteomic, and analytical metabolomic. So I like that. I've always liked that area. So advancing the technologies. I think some of our competitor journals tend to be more biology oriented. So we're like the bioanalytical side of analytical chemistry. And we love software papers.
Here we go. All right, and the other question that came through the Q&A, it's, so you mentioned, yes, that the best approach is to have one leading author, yes, and then the other will edit and correct it. But here we have a question. So I think so it's from a PhD student. Like if someone has especially a conflict with a senior author, like how that should be taken care of, if there is any golden standard there. if which advice to take, like what to do.
In terms of authorship?
I don't think so. It's about the authorship. I think so about how the manuscript should look like. Yes, which approach to take, because if you have several authors, as you mentioned, yes, there are different approaches, like how the manuscript particularly, should we go a little bit more biology, a little bit with the methodology, and if there are, for example, two senior authors, like how, and there is a conflict, like how would you advise them to deal with that?
Yeah, I think I would go with the senior author's experience on that. And so I've had discussions with people, and I'm usually right. just
How do you know that?
just the experience of having done this for a while is useful. Yeah, so I think experience counts for a lot. Having dealt with the system for a long time can be useful. Deciding on how to structure a paper and how to frame a paper to get the best to get it into the best possible journal. And I would also say that getting papers rejected, it's hard to not view this as bad and as something that's demoralizing. But some of my best papers have been rejected time and time again. And then there's a very famous scientist from Stanford, Ron Davis, who's a genomic scientist. And one of his former postdocs told me that he once said that his most highly cited papers were the ones that he had the hardest time getting published. And so sometimes you can be too far ahead of the curve and people don't get the significance of what you're trying to publish. And so then it can be a struggle getting the paper published because people struggle with it. And I would sort of a recent example of that would be the single cell work that Nikolai Slavov was trying to publish. I was a reviewer on one of his papers, and I thought it was great, but it got rejected time and time again from journals because people had a hard time believing the results. So you got to keep at it.
So do you think this is because the data themselves, or do you think it's the writing skill? More writing skill can help other reviewers to better understand this.
Yeah,
like I know your sequenced paper is highly cited. You also told in multiple times you have the story about the publishing sequenced. So now you were very young when you were relatively young, younger, when you're publishing the sequence. Let's say you had the similar results in the same time, but you have better writing skill. Do you think you can get it published in Nature or Nature Methods?
So, well, Nature Methods didn't exist back then. Not even sure Nature Biotechnology existed back then. The first place I did send it was PNAS.
Yeah.
And so I think what happened to that paper was I guess I can say it because the reviewer is no longer with us. So it ran into one particular reviewer. So when I sent it to PNAS, I sent it directly to Fred McLafferty as a National Academy member to transmit it to be the editor on it. And I think he reviewed it, and then I think he got Klaus Beeman to review it. In fact, I know Klaus Beeman was a reviewer because he admitted it to somebody. And so they rejected it. And then Fred McLafferty said, when you send it somewhere else, have the editor contact me and I'll tell him who the reviewers were, and that'll speed up the process. And that turned out to be a huge mistake because I then sent it to the Journal of Protein Science. And I told the editor that was handling it this information. I think he did contact that person because that person just kept rejecting the paper. And if you know Klaus, he had a short stint as an associate editor in analytical chemistry, but people complained about him rejecting every paper that came in. And so he didn't last very long as an associate editor. So Klaus had very high standards. So if the paper didn't come from his lab, it wasn't worth publishing, basically. So that wound up being a problem. And then when it went to a different journal where Klaus was no longer a reviewer on it, it got right in. But maybe some of the writing could have been improved. I think some of the issues that Klaus had with it, I think, centered around the fact that I was using a triple quadrupole mass spectrometer, and so it was lower resolution data. Klaus was an advocate of the a 4-sector mass spectrometer with higher resolution. It was the magnetic sector mass specs were the Orbitraps of the day. So the high resolution, higher mass accuracy instruments. But they had their own particular problems. They weren't very sensitive, and they were slow scanning and so forth. So Klaus was very much a proponent of that type of data, and so I think he had issues with the triple quad data that we were using.
Well,
Yeah.
It's happened if you happen to use, so it can happen to everyone. Yes, then. We are running out of time, but I would like to ask one more question. It's regarding the corresponding author. So, which should be the criteria for selecting who should be the corresponding author in a manuscript? Would it make sense for a postdoc to be the first and co-corresponding author together with PI, provided that they were the main responsible in the project, in the term of the design, follow-up, writing, etc.? What's your advice?
Yeah, I don't think there's a problem with co-corresponding authors. So oftentimes the criteria that I have for corresponding author is the person that's going to be in the same spot the longest, because like a postdoc, while they're here, they have a Scripps email address, and then they move somewhere else, and now nobody can get in touch with them. So oftentimes I look at it as, who's the person you can get in touch with to talk about the paper or to get information about the paper. So, but then using a co-corresponding author system can work too, because you have at least one person that's going to be fixed in position for a certain length of time.
Hey, John, I know you're running out of time, but I really want to ask you another question, last question probably. Now it's midnight in China, and there are over 2,000, close to 3,000 participants. You're very famous here. So you just mentioned one of your most productive or with the best writing skill is Dr. Meng Qiu Dong, is it? Meng Qiu?
Yeah, she was an excellent writer.
Yeah, so she's now in Beijing. Yeah, so I'm very surprised, and many Chinese students like me are very surprised because we're not so confident. English is not our first language, and we learn only in school. We don't use it every day. And when we start to summarize our data, we're always concerned about the English, and many of my students also have the same concern. So can you say a few words to the Chinese students? Let's say, how should they treat this inherent shortcomings in writing scientific papers? And can you tell some, maybe I can ask Meng Qiu next time, but can you tell us what's her secret to be
actually, I don't know what her secret is.
That's the writer in your lab. You have so many native speaker students.
Yeah, she's incredibly conscientious about those things. Like for example, when she got her job and she was going to get a mass spec. She was in a panic because she hadn't paid as much attention to operating the mass spec as she probably should have. And so she delayed starting her job by 6 months so that she could now go down to the lab and make sure she understood how to clean it and clean the mass spec and do all this other, tune it and do all this other stuff that she was not really paying that much attention to. And then she also made up this incredible list of every item that she needed to start her lab. And so she left a copy in the lab, and that's been passed along to other students. So Meng Chu was very conscientious about things. And so I'm sure she was agonizing, over every sentence that she was writing when she was writing it. So, but I think the important thing, particularly if English is not your first language, is that you need somebody to come in and correct your English, and not just go in and accept the changes that they've made, but understand the changes that they've made. So as a way to continually try to learn about sentence structure and verb use and things like that. So one of the common issues that my Chinese postdocs have made over the years is that they get the pronouns wrong, because you don't have pronouns in Mandarin. And so they're always messing up with the pronouns. So it's just things like that. And it's just when you are writing and you get somebody to correct the writing, some native English speaker, and there are lots of copy editing services out there now. ACS has one, for example. But when you get back that the thing with the changes, you want to look at the changes and use that as an educational process to understand, what did I do wrong here, and then understand it. So my wife does copy editing for all the papers in the lab, and there are certain people that make the same mistakes again and again and again, and they're not looking at what she changes and trying to understand why she's making those changes on their sentences.
Mm.
That's fantastic advice for any writer, I think.
Yes.
Maybe she can write a paper of the common errors for scientific writing. Write a book.
Well, I'll have my wife do that.
I guess we're 10 past the hour, so we've gone over quite a bit. I think, John, this was a fantastic session. We really appreciate you coming here and presenting this material and talking about JPR and your process as well. And with that, I think it's time for us to close. Thanks again so much for doing this.
Yeah, my pleasure. And especially for grant writing, you want a copy editor. You want to have other people reading your grants, so that things that you think are really clear may not be.
I think so. That's another topic of the discussion. Yes, how to write the grant. It's a little bit different process. So stay tuned.
So my recommendation for someone to do that is Ping. Pei is a fantastic grant writer.
Okay, here we go.
Maybe
Okay,
we
then.
can invite her to talk about this grant writing next time.
Yes, we make the on our wishing list this name and this topic. So one more time, thank you, John.
You're welcome.
Thank you, everyone, for joining us. And this webinar series is going to be continue every other month. So please stay tuned and have a nice day. Have a nice night. And one more time, thank you so much for joining us.